Welcome back to this next video, and this is the part 2 on the transforming growth factor beta. In the last video I've told you that the transforming growth factor beta, it acts through the receptor serine threonine kinase, and the receptor serine threonine kinase is made up of two subunits of the receptor. One is known as the receptor type 1 and the other one is known as the receptor type 2. So the transforming growth factor beta, it binds to the receptor type 2, and when it binds to the receptor type 2, it is actually going for the phosphorylation of the receptor type 1 at its serine residues, thereby making the phosphoserine in the receptor type 1. When the receptor type 1 it gets phosphorylated at its phosphoserines, it is going for the phosphorylation of the R SMADs, and when the phosphorylation of the R SMADs occur, the R SMAD they combine with another SMAD which is known as the SMAD4, and this complex of the R SMAD and the SMAD4 is moving into the nucleus of the cell, and in the nucleus they are going to act as transcription factors, thereby causing the transcription of those genes which are responsible for the G1 arrest in the cell cycle. Now in this particular video I want to focus on how this transcription it leads to the G1 arrest in the cell cycle. Now the R SMAD in this particular case are going for the transcription of a particular protein or a particular gene which is the P21 gene, and this P21 gene, when it is translated into the P21 protein, is actually responsible for arresting the cell cycle in the G1 phase. Now how this P21 arrests the cell cycle in the G1, let us discuss this in a little bit detail. Now for understanding the arrest in the G1, you first need to understand the normal condition, how the cell cycle is moving from the G1 phase into the S phase of the cell cycle. Now in normal condition, if you want to move from the G1 phase of the cell cycle into the S phase, you need a particular transcription factor which is known as E2F transcription factor. Now initially this E2F transcription factor is in interaction with another protein which is known as the retinoblastoma protein, and this retinoblastoma protein it can exist in two forms. One is known as the hypophosphorylated form of the retinoblastoma protein. I refer to the retinoblastoma as RB. So the RB can exist in the hypophosphorylated form. It can also exist in the hyperphosphorylated form. Now both of these you can say forms of the retinoblastoma protein, they are in the phosphorylated form, but in one case they are in hypophosphorylated form, that means they have less of phosphorylation, and in the other state they are in the hyperphosphorylated form, means they have got extra phosphate groups in the protein. Now when the retinoblastoma protein that is in the hypophosphorylated form, in that particular case it can interact with the E2F transcription factor, and when the retinoblastoma protein it is in interaction with the E2F, this E2F transcription factor is actually in the inactive form, and when this is in the inactive form you actually cannot move from the G1 into the S phase of the cell cycle. Now to release this E2F from the retinoblastoma protein, you need to convert the RB from the hypo into the hyperphosphorylated form. Now how this change is achieved, that is how the RB is changed from the hypophosphorylated into the hyperphosphorylated form. Now in normal condition, when during the G1 there is a particular cyclin dependent kinase which is known as the CDK4, and the activity of this particular kinase, as the name indicate, is dependent on the cyclin. So the CDK4 is actually a kinase whose activity is dependent on the cyclin. Now during the cell cycle, each of the phase like the G1, the S or the G2M, they have got their specific cyclins and their specific CDKs. So when you talk about the G1 phase of the cell cycle, the particular cyclin is known as the cyclin D, and the particular cyclin dependent kinase is known as the CDK4. So during the normal condition, the cyclin dependent kinase 4 it interacts with the cyclin D, and this complex is known as the G1-CDK. Now this G1-CDK, it acts on the retinoblastoma protein, and as this is a kinase, so when the kinase interact with the hypophosphorylated form of the retinoblastoma protein, that is converted into the hyperphosphorylated form. Now in the hyperphosphorylated form, the retinoblastoma, it cannot interact with the E2F transcription factor, and the E2F is released. Therefore the cell cycle moves from the G1 into the S phase of the cell cycle. Now what happens in the transforming growth factor beta pathway, in the last video I've told you that it's actually responsible for the arrest of the cell cycle at the G1 phase. Now during the transforming growth factor beta pathway, the SMAD proteins, they are going for the transcription of a protein which is known as the P21 protein. Now at the N-terminal of the P21 protein, there is a particular domain which is known as the CY1 motif. And this CY1 motif is actually binding to the CDK4. So when the P21 utilizing its CY1 motif, it binds to the CDK actually cannot interact with the cyclin D. Now what this means is that this is a cyclin dependent kinase, and if cannot interact with its cyclin, that means it's remain in the inactive state. And when the CDK4 in the presence of the P21 cannot interact with the cyclin D, it cannot actually convert the retinoblastoma protein from the hypophosphorylated to the hyperphosphorylated form. So in this case, the retinoblastoma that remains in the hypophosphorylated form, and when it is in the hypophosphorylated form, it will be in interaction with the E2F transcription factor. That means that the E2F will not be released, and your cell cycle is going to stuck in the G1 phase of the cell cycle. So if you like the video, please subscribe to my channel, hit the like and the notification bell, and share it with your friends.